So some basic information on the rose in general. It's kind of a quirky crop even within the horticulture world. Rose can apply to anything within the genus Rosa, and depending on who you ask, that can be between 100 all the way up to 200 different species. I usually stick with the mid-range estimate of about 140 different species. The genus can have a wide range of ploidy levels, all the way from diploid to hendecaploid. So something actually what I have been working on so far is determining the ploidy levels of some different germplasm that we have so that we can not try to cross a hendecaploid with a diploid or something like that. Modern garden roses, however, what you'll see planted around campus or what you can pick up at Lowe's or Home Depot are not usually these species. They're usually interspecific hybrids. Most of them trace to about 7 to 10 different species and not the rest of the germplasm, the rest of the genus. So it's a very narrow subset of the genus that's actually used in breeding. And since these are hybrids, they're usually highly heterozygous, which makes genetics a little bit interesting. So this is a phylogeny of the rose genus based on chloroplast sequences. And I know it's not really legible. It's not really legible up here either. But the main point is the stuff in green here. These are what I've been calling the big seven, the main rose species that have contributed to the modern rose. So these are Rosa gallica, Rosa moschata, Rosa odorata, Rosa wicheriana, Rosa multiflora, Rosa chinensis, and Rosa fotida. And the important thing here is that they're all sort of clustered down here on the bottom of the phylogeny. This paper at least split the genus into two main clades, clade 1 up here and clade 2 down here. All 7 trace to clade 2, and this will become important later on when I'm talking about my project specifically. So currently there are over 20,000 cultivars, but this is not only garden roses. Roses are also bred for floriculture to get long-stemmed roses for, say, Valentine's Day. They're bred for the perfume industry, for culinary purposes. You can make tea out of the petals and out of the rose hips or the fruit, and also for medicinal purposes. However, when it comes to garden roses, there are certain traits that are very, very desirable. My advisor, Dr. Byrne, did an informal poll of growers and rose enthusiasts, and they found that #1 trait desired is disease resistance. It blew out all the other traits out of the water. Roses get a lot of different diseases, especially fungal diseases, black spot, powdery mildew, depending on where they're planted. Other desired traits include novel colors, good fragrance. A lot of modern roses are accused of having poor fragrance, and this is in part because the gene for fragrance, for the classic rose fragrance, is linked to a gene for disease susceptibility. So as we breed for disease resistance, we tend to lose the fragrance. So that is something that certain breeding programs are working on. And then there are a couple of traits that are considered standard in roses and that people just assume is going to happen. That they'll be ever blooming, they'll bloom throughout the entire growing season, and that they'll be double flowers. However, the problem is that most of the rose species, of those 140, do not have these traits. Most of them are once blooming. They'll bloom once in May or June, then be done for the rest of the season, and they're just a green plant sitting there. They also generally have single flowers where there are only 5 petals, and you don't have these huge lush flowers that you can see on shrub roses around campus. Some background on the disease I'm working on. It's called rose rosette disease. It's a fairly new plant disease. It was first observed in 1941 simultaneously in Canada, Wyoming, and California. However, since then, it's moved eastward and is now mostly centered in the central and eastern United States. So if you look at the map here, some of the early states are on the west coast, but most of the dates are on the east coast. What is interesting to note is that we don't actually have rose rosette disease here in College Station yet. If you draw a line a little bit south of Dallas all the way across the continent, we don't see any rose rosette disease below that line, yet at least. We don't know why this is. It could be that that the disease just doesn't survive that far, but there are other factors involved with the disease that I'll get to in a moment. It wasn't until 2011 that the causal agent was identified. It's a virus. It's called rose rosette virus, and it's tentatively been put in the genus Amaravirus. Amaraviruses in general are not very well understood. It's a relatively new genus and hasn't even been assigned to a family yet. But they are all negative sense single-stranded RNA viruses, and they all have in common that they're mite transmitted. So rose rosette disease is transmitted by an airborne mite called Phyllocopies fructiphilus. And the virus has its reservoir in a rose called Rosa multiflora. Rosa multiflora is a non-native rose. It's actually highly invasive. It was planted heavily in the 1940s and 50s as a method of erosion control and as a living fence, and the planting of it was highly encouraged. Since then, we have discovered that it's very well adapted to parts of North America, and it's now considered a noxious weed in many parts of the country. It's also highly susceptible to rosette disease. So it'll get infected, but it doesn't die fast enough to cut off the reproduction. It'll still spread its seed, and it'll continue to spread and be a noxious weed, and then the parent plant will die. So we don't really get rid of Rosa multiflora with rosette disease. So some other factors that could be contributing to the fact that we don't have rosette disease here could be related to Rosa multiflora or to the mite that transmits the disease. We don't get a lot of Rosa multiflora in this part of the country, so it could be that just the virus, there isn't enough virus present here to have it be a problem. Or it could be that the mite that transmits the disease just doesn't like the warmer weather, and we don't know what is actually responsible for that. Another problem with rosette disease is that the symptoms vary with the cultivar and with the stage of the disease. However, by far the most widely publicized symptom is what's called the witch's broom. This is usually what it looks like. It's a red fleshy growth that's more or less broom-shaped or sometimes described as rosette-shaped. And you could see multiple of these on a single plant, or there could just be one. But there are other symptoms as well. An unusual red color could be a symptom. New growth on roses usually is red in color, but if that red color sticks around for weeks or even months, it could be a sign of rosette disease. A succulent stem, as opposed to woody growth, could also be a symptom, and that may or may not occur with the red color. Thorn proliferation can also be a symptom, going along with the sort of excessive growth that's found with witch's broom symptom. A lot of roses are naturally extremely thorny, but if, for example, you know that your rose isn't one of those types, and all of a sudden you see new thorns and more thorns on new growth, it could be a sign of rosette disease. Rosette disease can also affect the blossom, resulting in blossom blight. And eventually what we can get is a dieback. As the rose is afflicted, it doesn't do very well in winters, and so in the spring they might look something like this. There doesn't look like there's much of a rose there anymore, and that's because there really isn't. Rosette disease is usually fatal in 2 to 4 years, depending on the cultivar. And so it's a huge problem because there are no consistent control methods. People have recommended pruning out the witch's broom or spraying for the mite, but really the only control method we can absolutely recommend is digging up the entire diseased rose, bagging it, and throwing it away. That's really the only thing that consistently works. However, on the plus side, it is exclusive to roses so far. There have been attempts to inoculate other members of the family, of the rose family, with the virus, and it hasn't worked yet. However, most cultivars are susceptible to the disease, maybe even all cultivars. We've been working with Jennifer Olson at Oklahoma State University, and she's been monitoring the Tulsa Municipal Rose Garden, where they have extensive rose plantings and very high rosette disease pressure. So this is a summary of some of her results. On the left column, we have different classes of roses according to their growth habit. What's interesting here is that something like hybrid tea, which is the third row, they have a stereotype of being very susceptible to diseases and generally wimpy. Shrub roses, for example, though, have a reputation for being very hardy and very disease resistant. And what we're finding is that when it comes to rosette disease, that's really not the case. Hybrid teas, shrub roses, all these different classes of roses will get the disease. Moreover, just because we have an asymptomatic category over there where the plants haven't shown any definite symptoms yet, doesn't mean that those are necessarily resistant. They might have just gotten lucky, or they might be harboring the virus and just haven't shown symptoms yet. So something that we're working on is narrowing down that list of asymptomatic roses to see if there are any truly resistant cultivars. So far we have one very promising cultivar, but everything else is proving to be susceptible. On the plus side, though, we do have several species from those 140 members of the rose genus that are reported to be resistant. This is mostly based on grafting trials or inoculation with the mite. However, we don't know anything about the inheritance of this resistance. The title of this talk was originally going to be the search for quantitative resistance to rosette disease, and then my advisor pointed out that we don't actually know that it's going to be quantitative. We literally know nothing about it. So I had to change the title. But the species, we have some diploids and some tetraploids that are resistant so far. These are Rosa satigera and Rosa palustris for the diploids, and for the tetraploids, Rosa carolina, Rosa californica, and Rosa spinosissima. I skipped over Rosa bracteata because it's not truly disease resistant. It appears to be resistant to the mite that transmits the disease. We can infect it with the virus. However, in nature, the mite doesn't feed on it. We're not really sure why. It might have something to do with the presence of trichomes around the mite's feeding area, but we're not really sure about that. So there's a definite need for rose rosette disease-resistant cultivars, unless we want to plant these 5 species forever. And that's not really a good idea, considering that people have those ideas for what a rose should look like, that it should be ever-blooming and it should have double flowers. These are all pale pink. Most of them are pale pink with our 2 white species in there. They all have similar growth habit, and they sort of grow over things and not a nice compact shrub. They're single flowers, and they're kind of cute if you have one of them, but if that's all you have for roses, it gets a little bit boring. So we definitely need rose rosette disease-resistant cultivars, and our approach is going to be through marker-assisted selection. Our goal is to find QTLs for resistance, and use that to speed up the breeding process. To do this, there's actually a nationwide project. It's called Combating Rose Rosette Disease, and it has 5 aspects. Best management practices, where we're trying to figure out the best control methods for the disease. Diagnostics, they're trying to come up with a reliable field test that you can do on the spot to see if the virus is even in the plant before it's showing symptoms. Breeding and genetics, which is where I am. Extension, getting the word out there, especially to landscape companies who will sometimes plant roses, they get infected, and then no one really does anything, and they just serve as another virus reservoir. And then marketing and economics. We want to find out in part whether our marker-assisted selection approach is actually going to be economically viable, and whether people are actually going to want to buy the roses that we create. That's kind of important. So the first thing we're going to have to do for our QTL analysis is the creation of segregating populations. This is a little bit complicated by the fact that we don't know anything about the inheritance of resistance, but fortunately we have some help. We're collaborating with breeders nationwide who are making crosses for us and sharing what they find. We have 2 resistance sources that are going to contribute to our populations. These are the diploid species, Rosa palustris and Rosa satigera. Last year, we made a number of crosses with Rosa palustris and a few with Rosa satigera. And what we found is that Rosa palustris is a really terrible parent. This is a summary of some of our best crosses involving Rosa palustris last year. OB and EB with Rosa palustris just indicates which form. It's a once-blooming or an ever-blooming species. And so we were keeping track of which one was which. The best crosses resulted in 5 seeds per pollination at the most. A single pollination in rose can result in 5 to 25 seeds per pollination. So that in itself is not that great, but we could make it work. However, on average, a cross with Rosa palustris produced less than 1 seed per pollination. So that's not very promising. There's also indication that, strong evidence that most of these seeds probably won't even germinate. We don't have germination results yet. The seeds have to be stratified for quite a while, and they should be germinating soon. But based on embryo rescue results, probably most of the embryos are not even going to be viable. So we're going to have to turn to another resistant source. We're not giving up on palustris, but it's not going to be the emphasis. So we're turning to Rosa satigera, which, based on the crosses we did last year, is going to be much more promising, and this is probably why. This is the phylogeny again, and here are the big 7 down here. Rosa palustris is all the way up there in clade 1, an entirely different part of the genus. So there's a great deal of distance between it and most of our rose germplasm, which traces to those 7. But here is Rosa satigera, clustered right down here with the big 7. So it's much more genetically similar, and what we find is that there are actually breeders who have successfully used Rosa satigera in the past. So we think we'll probably be able to use it as well. So this is Rosa satigera here, a close-up of the flower and the plant overall. It's kind of a cute-looking flower, but it still has the same problem that it's a single flower. It's just pale pink, like pretty much every other species rose out there. And the plant type is sort of rambling, and it likes to climb over everything, which is proving to be a bit of a problem in the greenhouse, where it's sort of trying to take up a lot of our space. However, it's diploid, it's closely related to everything else we have, and there are already hybrids out there. We don't know if these hybrids with Rosa satigera are resistant to rose rosette disease, but it's a promising approach, and so we're also going to try using those as parents in our population as well. However, Besides the fact that it sort of wants to grow over everything that we have, it also has the problem that it's what we call cryptically dioecious. It is the only rose species that we know of that is dioecious, and it just happens to be one of the ones that's resistant. However, looking at the plants, you can't tell if it's male or female. We know that some of our accessions are female. They've already been tested, but most of the Citigera that we've collected and that people have been sending to us, we have absolutely no idea. So before we make any crosses this spring, something that I'm going to be doing is called a hanging drop pollen assay, which is where we try to germinate the pollen. If it germinates, it's viable, it's going to be a male plant. Because Rosa Citigera is very definitely male or female. You don't really get any sort of in-betweens. So I'm going to have to do that before we make any crosses this spring so that we don't repeat what we did or didn't do with Rosa palustris. So our ultimate goal is that when we have these populations, it's going to be 6 interrelated F1 populations of at least 80 individuals apiece. And this is sort of what it's going to end up looking. This is a highly simplified version. I tried to put the full version of the crossing scheme up here, complete with pedigrees, and then I realized it wasn't legible even on a computer screen. So this is a simplified version. And the idea is that we have 2 different resistance sources. Here's Rosa palustris, and here's Rosa Citigera. They are both being crossed to the same 2 cultivars. So we'll have a population here and a population here that are that have resistance from 2 different parents, but they have the cultivar in common. So that's what I mean by interrelated. The 80 individuals seems sort of arbitrary. We're shooting for a lot more than that, at least 100, but we think we might be able to get away with 80. Hopefully more than that, though. Once we have these populations, we're going to send them off for phenotyping. Since rose rosette disease isn't here in College Station, we can't do that phenotyping here. So we're collaborating with Dr. Mark Wyndham at the University of Tennessee for our rose rosette disease phenotyping. And this is a diagram of their planting there. So the important part for this talk, at least, is stuff over here. The arrows indicate the direction of the prevailing wind. This is a row of infected plants that are also infested with the mite that transmits the disease. And then these are the plants, our progeny, that were having phenotype for rose rosette disease. So the idea is the wind will blow these mites that are carrying the disease into our progeny, infect them, and we'll see what lives. And so this phenotyping will actually go on for 3 years. That's pretty standard for roses to have a several-year trial for disease resistance. We will be able to do some phenotyping ourselves. We'll have plantings also in Overton and in College Station to phenotype them for horticultural traits. So this is going to be our standard phenotyping, which involves things like flower quality, flower color, notes on the fragrance. We'll rate it for the overall appearance of the plant, whether it's sort of scraggly and ugly or whether it's making a nice, well-rounded shrub, and how much of the plant is covered with flowers. We usually want a plant that's at least 50% covered with flowers. And as well as ratings for disease resistance to other diseases that we do get here. And we get the usual fungal diseases just like any other part of the world. So that College Station population will provide our source for DNA for all of our QTL analysis. We'll be using a method called digital genotyping for our SNP identification. We've decided to use SNPs because they are relatively easy to get a hold of, to create, and then also we can get a lot of them in one go. The method we'll be using is digital genotyping. It's a high throughput genotyping method that was developed by Dr. Trish Klein's lab for sorghum, but it does work for roses as well. Our goal with that is to get a minimum of 1,000 informative SNPs. Part of the problem with digital genotyping, though, is that it requires the use of a reference genome to compare these fragments to the reference genome and see where the polymorphisms actually are. And we don't actually have a rose reference genome. When I came here, they told me we'd be getting a reference genome soon. They're still telling me it's coming soon. So we'll see when that actually gets there. In the meantime, we're using the strawberry reference genome, and there actually is good synchrony between the strawberry genome and the rose genome, so it works. We'll be creating a linkage map using the join map software, and this will be a consensus map that involves markers from my rose rosette disease populations, as well as SNP markers from my lab mate, Muchin Yan's populations. She's doing QTL work on black spot resistance, a different disease of rose. And we'll be using SSR markers from a pre-existing consensus map to link these maps to what we already know about roses. Our QTL analysis itself is going to be what's called a pedigree-based analysis. This is pretty much exactly what it sounds like. We use the pedigree to help inform our QTL analysis and our later breeding decisions. Wow, I just lost my train of thought. So this is going to be a bit of a problem for me because most of my resistance sources are species, which by definition have no pedigree. So my pedigree is usually dead end very quickly. However, we might still be able to get some advantage out of this, and if not, we'll still be able to find QTL eventually. The idea with pedigree-based analysis is that you can trace where the resistance alleles actually came from all the way through the pedigree and inform your breeding decisions better. We do this through two different software, PediMap and FlexQTL. PediMap was where that pedigree and crossing diagram I showed you earlier came from. It's good for making pedigrees in highly heterozygous and outcrossing crops, which is sort of rose in a nutshell. And you can also combine genotypic and phenotypic data in that same pedigree, so you can trace alleles and even individual markers or haplotypes all the way through the pedigree. FlexQTL then is going to be our QTL discovery software. So we're going to feed all of these populations into FlexQTL at once. And the idea behind that is by looking at multiple populations simultaneously, we can improve the QTL detection accuracy. This will also hopefully validate the QTLs at the same time, so that the QTLs and markers that we find are actually relevant besides our specific germplasm. That's a big problem in QTL analysis, is that you find a marker in a QTL in your population or your germplasm, and then it doesn't really work anywhere else. So we'll also be bringing in germplasm from our collaborating rose breeders and test out these markers and QTLs in their populations as well, and further validate the QTLs. Because the ultimate goal is not just to find the QTL and then have me graduate, but also in the long term to actually use that QTL and integrate it into the rose germplasm to create rose rosette disease resistant roses much more quickly than we would otherwise be able to. So I'd like to thank my committee members, Dr. Byrne, Dr. Klein, Dr. Pemberton, Dr. Ong, and Dr. Stelly, and then the people who have been helping me with chromosome counts and the creation of my population so far, our research associate Natalie Anderson, and then Dr. David Slezak and Dr. Bo Liu. And I'll take any questions now. Yes.
What species is the knockout rose, and is it also susceptible to rose rosette?
Knockout rose is susceptible. It's no more susceptible than other cultivars, but yes, it traces. I think it traces back to Rosa multiflora, but I'm not actually sure about that. So that would explain why it is susceptible.
And then if it takes a long time for symptoms to sometimes show
Yes.
up, how will you do your phenotyping? Will you use the diagnostic test that's being developed to see if virus is actually present? Because, you know, if you get some symptoms showing up in some of your plants really quickly and then others it takes a year or more, how do you...
Yeah, so one approach would be to use those diagnostic tools. Another is that for the ones that aren't showing symptoms in the field, I believe the idea is that we'll then take them and graft the disease into them, and hopefully they'll speed up the process of them showing symptoms. But yeah, that could be a potential problem where something gets pegged as resistant when it's really not. So, yes.
So at the end, you want to breed something, some rose type that resists infection of the virus or do not show symptoms or both?
Preferably doesn't get infected, because if it just doesn't show symptoms, then it could be a reservoir of the virus. But if it's both, you know, that's okay too. So. Yes.
So does the virus need mice to be infectious, or just like for cattle?
Wait, say that again?
So the virus, does it need mice to be
spread?
infectious, or?
Yeah, it does.
Only might transmit it, or can it be?
There are other vectors.
Right.
Other-
It can be transmitted by the mites or by grafting infected plant onto healthy plant.
Mechanical infection.
Mechanical, as in like through like pruners or something, that doesn't appear to work at all. It has to be through grafting or through the mite.
Okay.
So. Yes,
DNA or RNA virus?
it's an RNA virus, single-strand RNA virus.
Is your level of infection adequate for screening, do you feel, by allowing mice to transmit the virus? Or do you visualize actually making something like a infectious transcript to make sure your inoculum load is the same amongst your population?
Oh, I don't know. I don't think we've.
I'm just curious.
Yeah, I don't think so. I don't think we've thought about that at all, or at least I haven't, so I don't really know.
I had the same problem in my master's because the wheat crawl might transmit the wheat mosaic virus, and I saw a lot of the literatures, they were either putting the same immunosuppressor, or and then the ratings was done based on the percentage, how much severity and how much infection percentage.
That might
And
be.
I did another way, resistant and susceptible, just by looking under the microscope. And then I got a feedback that I used to do, like doing the severity percentage. I had the same immunosuppressor, but luckily either way I found the same QT locus.
Okay.
That would be helpful.
There's a suggestion, maybe, because like I explained to us, it's 3 years of screening, long time.
Yeah.
For, at least for us, from crops. So maybe you spend some time grafting every plant in every location. I don't know how many plants there will be, but
Yeah, that is something that.
just to ensure.
Yeah, we're working with, like, our collaborating members of the project are doing more of the grafting. I probably won't be doing the grafting myself because that would involve bringing in the disease here. But yeah, I think that is something that is in the works. Maybe not for all of them because that'd be a lot of plants, but definitely for some.
Yeah, when I was doing a postdoc at Kentucky, I was working with a virus, and we could make infectious transcripts and then use carbon and rub it on the leaves so that you could have a known amount of inoculum, and that would rough up the leaf, and then it would get in that way, and then it would go systemic. I'm just curious if anybody. I know you have. There's a lot of people that work with the actual RNA virus that are developing the diagnostic test, but I wonder if they have any information on the infectivity of the RNA virus as an infectious.
Yeah, I don't know. I haven't heard anything about that. I think I'd have to ask them and see what they know.
So that's what we do with tobacco itch and tobacco mosaic viruses is make a buffered solution that lasts for 5 or 6 days and go through a lot of Q-tips.
Yeah, I don't think it'd be quite that simple because you can't transmit it like through
pruners,
or anything like that. So I'm not sure how you do that. It was injected somehow, but yeah.
I have a question. You've shown us the picture of Rosa setigra.
Yes.
The rose doesn't look attractive.
No, it's not very nice.
So, do you see, in these crosses, do you see like good-looking roses?
Well,
setigra?
we haven't seen anything.
Because if you don't
yet.
see that, what's the point?
We don't have any germinating seedlings from these crosses yet.
Okay.
But the idea is that even if the first round that I'm actually going to be doing my QTL analysis on, if the F1 population doesn't have those traits, we can hopefully back cross it to one of the cultivars that is much more attractive and get those traits back as well as keeping the resistance.
Yeah.
So.
My point is that if the species you're using to get resistance does not give you the phenotype,
Yeah.
the rose phenotype you look for, there is no use in using it. I mean, it's just...
Yes, that is a problem, but we don't really have an alternative. There are no... The one cultivar that looks promising for actually being resistant is actually a tetraploid, and so we keep the diploid and tetraploid breeding programs separate, and so I can't actually use it. So it wouldn't actually be much good for this. But
Could you not double your diploid?
you could, but that's not really part of the plan. We could try that, but I don't know.
8 year PhD.
Yeah, no.
The coffee group did that, where
Yeah.
they took a diploid and they colchicine and they doubled it,
Colchicine, yes.
and then they crossed it with a tetraploid, because Arabica is a tetraploid. So I was just wondering if you had like a diploid that had like a tetraploid that had really nice traits, and your diploid that has nice phenotypic traits, but it's your tetraploid that has the resistance. Would there be any way of doing that kind of a
Yeah,
cross and then seeing if you can develop something
that
that
would-
has, because then you'd be bringing good phenotypic traits essentially from both
yeah.
sides. I don't know.
That would be a possibility.
I don't know how easy that would be.
Yeah, I know it's been done in roses before, but I think it really slows things down a lot. So
Yeah.
I don't know.
I have one comment, resistance to the virus and to the mite are
Yes.
different things. And, from experience with wheat, sometimes you see highly resistant lines to the mite that have a lot of virus. And some lines that have no resistance to the virus, to the mite, but they have good virus resistance. It's very complex. So when you map those QTL, you might need to look for QTL that provides resistance to the mite per se, and then QTL that provides resistance to that virus
Yeah.
separately.
Yeah, that's a good idea.
Well, that's why the grafting would help with resistance to the virus screening, because then she knows that it would be,
The
it's
virus.
resistant, it's due to the fact that it's the virus resistance, not the mite, because she won't necessarily know if the mite's feeding on her plants or not.
That's right, and that's why
Tennessee.
they might need to use like small cages, because they're
Right.
using now rose and wind to blow the mites to the
Right.
lines. That might not work very
Right.
well, actually.
Right.
So you need to
They're
be careful.
microscopic though, I'm assuming, right?
Yes, they are.
Yes, they are.
I don't know what to use, but
Since
you
we're
need
all
to
Scotch tape.
brush tape, whatever.
Yeah.
Let me ask this. From a breeding viewpoint, are some of your susceptible parents to which you're crossing, are they agronomically acceptable?
Yes.
Okay. 'cause I
Yeah,
wanna-
they are.
because a
They're
lot of times you-
they're standard rose cultivars that
Okay,
people
so
like.
you already partially there, if you actually do find some of your F1s that are resistant,
Yes.
where you could back cross to your acceptable. Because if you
Yes.
try to move it into a completely different background, I think many of us that have worked with QTL, like all of a sudden those QTL disappear
Yeah.
out of the background. So
Yeah.
that's good. I know nothing about viruses. It seems like the group does. Is this a typical phenotype for a viral infection? The proliferation, the growth with a virus? I mean, I know nothing about
Seems
viruses.
a little unusual, but, I mean, a lot of times, you get like fluorosis and curling and.
Broad mites will cause damage like that, but I think it's from a chemical they ingest,
Yeah.
not a virus.
Physiological.
You could just change
To the
the,
extent of whether you're here for 3 years or 10 years, as Bob suggested, you mentioned lots of populations. Do you have an idea of how many populations? How many individuals per population?
Yes,
And if there's any chance to take a shortcut, if this might be a major gene, can you take bulked segregated approaches instead of entire QTL population?
We're hoping for 6 populations, each of at least 100 individuals.
All done by you.
Oh,
And the collaborating
other
breeders.
student, Mandy's doing 15 populations
Yeah.
of like 70 to 80.
Yeah,
Like,
so-
Farming out the SNP work, right?
What to us, I mean, we're doing it with her, but she's already
You're on
got a
track
bunch
for
of them
6 years,
done.
not
It's
3 years.
crazy.
It's going to be at least 5 years, yeah. So,
And is it a major gene you can tell?
we don't know.
But that's the
we
problem, right?
know absolutely nothing about it, so we just know it's there.
Wow.
Something is there.
So has the White House rose garden been hit?
that's where
I
it
don't
gets
actually
fun.
know. It's definitely in that part of the world, but
We could take it over there.
it's true. Yeah.
Really create some recognition for that disease.
Back up to your diagram from Knoxville,
The which one?
diagram from Knoxville. That one.
That one. There. Okay.
The red is the
Infected versus-
infected ones. Why is the first series going to be blank except for one end of it?
That's where we don't actually have anything planted yet. So it's just empty. It'll probably get
You're
filled
going
in.
to plant, okay.
Yeah.
So there's no
There's no-
design rationale for that.
Right. It's just there's literally nothing planted there yet, but there definitely will be.
And do you know if the ones in series 1, 2, 3, 4, 5, in the 6th series away from the infected plants, will the mites distribute as well in that series as they do the first series?
Possibly not. So they're also going to supplement with plant by interspersing infected plants in there, or by bringing in cuttings of infected plants and grafting them on, or just spreading the mites manually if necessary in there, just in case the prevailing winds aren't doing it. So.
Do the
They
mites,
expect to be a
Mechanically
problem.
disturb those, the infected plants to make the mites move?
Well, since they're airborne, it's really not up to the mites, but we could they are going to.
the blower?
They are planning on putting up fans to help with that.
So how come you didn't do a buffer row all the way around, I mean, an infected?
Yeah, exactly.
Thing
Good
all
point.
the way
I'm sure
around?
this-
Prevailing wind.
I'm not sure
That's
what
a
the
good idea.
Because I
idea
know
behind
prevailing
the design
wind,
was.
but I mean,
Yeah, they need to have it all around us.
it just
Yeah.
seems like if you were gonna have fans, you could be having it come in from all.
This part wasn't up to me. I'm not sure what their motivation was for this exact design. It might have just been they didn't have
The
the
guy
space
doesn't
for
want
it.
those mite plants any closer to the rest of the.
Yeah.
The whole thing. Although this is all actually going to be planted as roses for rose resent disease, so I don't know why you wouldn't plant even more disease plants. But I guess the idea.
Gotta be something over here on the right-hand side, unless it's the Tennessee River.
I do not know.
Can the mites survive independent of the rose? Like for rearing purposes?
I don't actually know. I don't think anyone's actually.
You
tried
can't
that.
cultivate or like grow mites.
You know, from my experience, it needs the plants to survive. Otherwise, within 24 hours, it dies.
The mite.
I would believe that. Yeah.
So what happens during the winter? Does it kill? Do freezing temperatures?
In wheat, it survives the winter.
Yeah.
Like it stays inside, and then once it's spring, it comes back.
Yeah, in roses it overwinters, usually in the plant, well, not in the plant, but hiding sort of underneath in the buds. That's how it lasts the winter. Yes.
Cut all
So
the
the
buds off.
question on that point, two things I don't know, and maybe you know. Center of origins for roses, and which agency keeps the germplasm bank for that?
Center of origin for roses, they're native all over the northern hemisphere. Most of those big seven that I was talking about come from Asia and Europe. Rosa palustris and Rosa satidra, though, are North American species, and they're found all over the continent. As far as germplasm, there's no collection. There's no germplasm bank, no.
So where do you get germplasm from then?
Collaborating rose breeders. And rose enthusiasts who see a Rosa palustris plant growing by the side of the road, take cuttings and send them to us, that sort of thing. So,
Be on the lookout.
all right.
All right. Well, if there's no other questions, let's thank her for her
Good luck.
presentation.